group, and he's going to tell you a little bit about his work and what he's been doing in the last.
Thanks for giving me the opportunity to present my work, and thanks all for showing up in these apparently busy times during applications and things. So the reason I'm here is because of my Marie Curie, which is a 2-year project that I have in the Netherlands as a postdoc. But then within this, you can have this secondment of 3 months. So that's in the grant I applied to work with Andy and Victor and Maria Jose and people here during this project. And basically what I'm interested in is species movement in aquatic ecosystems. So that's also the reason I'm here working with you. But also the reason I'm here is with the aim to interact more with people working on species movement in terrestrial ecosystems and to see if we can set up some more collaborations and things. So if anyone is interested to talk about what I'm now saying here after the talk, then I'm still here until the 15th of December and I would like interactions. So I'll first give a bit of an overview of the things that I'm generally interested in and then go into more detail about one of the topics. So what I'm studying overall in all kinds of projects is the interaction between species movement and species interactions. So generally species movement, for instance the flight of a bird from one site to another site, leads to new interactions between this bird and other species. But the opposite is also true, that a bird might be flying and might be interacting with species such as plants during their movement. So I'm studying both how movement leads to species interactions and how species interactions can cause movement. And the reason I'm interested in this is because, well, as humans we are doing all kinds of things to the natural environment. We're destructing it, we're warming it up, we're polluting it, and we're fragmenting it. And on top of all, we're putting all these alien species in the aquatic systems particularly. If we want to know a little bit more about what species may be able to cope with these changes, we need to know which species are able to move and what they will encounter if they move. So this is my motivation for studying these kinds of things. I study this in aquatic freshwater ecosystems, ranging from fens to rivers to ponds and lakes. And basically the 4 components that you can divide my work into is one of the things is studying if a species arrives into a new community, for instance a plant community, what happens. So with a PhD student in the Netherlands, Antonella Petrozzella, she's doing work on the biotic resistance hypothesis, basically stating that if an alien plant species arrives into an existing plant community, how important the species richness in this plant community is for the alien to establish. So that all has to do with facilitation and competition of plant communities. And also, if you try to restore an aquatic system, what kind of plant species should you deliberately introduce to accelerate this restoration? So that's basically a plant community ecology part of my work. But then also I've spent some time in Norway working on movement of animals, particularly fish, in response to fragmentation of river systems. So as humans we try to generate this green energy these days, which is very hot, which I totally support. But on the other hand, we're building these large hydropower dams in rivers, which blocks the possibility for fish to migrate past these dams. So I did some work on how this river fragmentation is affecting the population genetics of migratory fish species and the efficiency of fish passages that we make when we build a dam, that we realize the fish cannot longer pass. Then you build a little stream on the side that the fish can still use. So this has to do with habitat fragmentation and animal movement. And then it gets even more interesting when you start to look at plant-animal interactions. So P.U. Zhang is a PhD student in the Netherlands, and he recently graduated on the effects of global warming and eutrophication on herbivory, so animals eating plants. And he found that if the water temperature of aquatic systems rises, that the metabolism of animals generally goes up, ectothermic animals, which increases the grazing pressure on plants. So plants will become more pressured by these animals eating them. But at the same time, there's eutrophication in these systems where the nitrogen content particularly goes up in the plants, which also makes them more palatable for animals. So animals also increase because plant quality relative to animal quality goes up. So that's part of plant-animal interactions. And then the topic I want to talk about more today is what we're doing with Maria José here. And that's basically what I started with as well, so during my PhD. And I'm still working on this topic, and that's dispersal of aquatic organisms by more mobile animals, such as particularly waterbirds. So we know that both plant seeds and invertebrates are being carried by waterbirds that are flying from one side to another side. And they can transport both internally after eating, but also externally by attachment, all kinds of aquatic life. So that's what I want to go into more today. But basically, I wanted you to know that I'm studying all kinds of different things. So if you're interested in discussing anything about this, then more than welcome. But the focus of today is the species movement in aquatic ecosystems, and particularly by animals and birds. And this process is called zoochory. So chory means movement, by zoo by animals. Because the question is already very old: how do all these aquatic plants and invertebrates that seem very immobile by themselves, how do they manage to colonize all these remote areas? Well, of course, some animals can just swim or jump or fly between aquatic systems, but others rely on this passive movement. And that's the zoochory comes in. So all kinds of animals can transport both internally and externally small aquatic organisms. And this is a picture of ectozoochory, quite drastically. This is lamna, the duckweed, being attached to a duck. And if you imagine if it flies from one side to another, then the duckweed may be re-entering the water. But also endozoochory, this is called internal transport by animals. And that is when you collect droppings and you find all kinds of seeds and invertebrates in these droppings that have been passing through the digestive system of birds alive. A brief overview of how this research field developed, large part thanks to people working at EBD. But basically Darwin was the first to see the discrepancy or the relationships between the wide distributions of these species that seem to be very immobile, and started to do work on external transport, which was realized to be maybe very important. So people started reporting sights of a bird flying with snails on its back, and we think, hey, maybe the snail has been carried. And then at some point it was followed up by experiments where you deliberately attach invertebrates or seeds to feathers, and you hold them in the wind flow, and you think, how long can these be remaining attached? And then followed up by field observations, basically catching birds and seeing what's on the outside. Internal transport was also some anecdotal observations, finding some droppings in the field of a bird and seeing that there are some seeds or invertebrates in there. And then quickly followed by all kinds of experiments where you deliberately feed a species of interest to a bird, and then you sit back, relax, and wait for it to defecate. And then you can estimate whether or not this species can be dispersed by birds, yes or no. And then field observations is also more recently. And that's basically collecting droppings, which we are doing quite extensively still. Droppings of birds and then seeing what's inside. And then more recently also distribution analyses, trying to find relationships between the distribution of plants, for instance, and the movement of animals. And this includes modern work with GPS tracking of birds, and also genetic studies to see relationships between movement of animals and the stuff they might be dispersing. And this is a rapidly growing field. If you just look at the number of aquatic invertebrate taxa that we know of that they are dispersed, and over the development over the year, you can see that the more studies we do, the more we realize that many invertebrates can be dispersed, and this is already quite outdated data, I guess, because we know a lot more these days. So today I would like to structure the talk with about 4 questions, like which species can be dispersed, which birds may be important vectors for these invertebrates and plants, how far can they actually be carried, and do we see any effects on genetic diversity of the dispersed species. First question, which species can be dispersed? Well, then we need to know what they will go through when they are dispersed, because For external transport, what you need to tolerate as an aquatic species is to be desiccated, because you're on the outside of a bird and you're flying in the air, so you won't have access to water. You also need to be able to stick to the outside, so you don't want to fall off. So these are two tolerances that you need as a species to be able to transport outside of a bird. For internal, the list is quite a little bit longer, so you need to be able to resist the high body temperature of a bird. It's about 42 degrees when you're inside. There's no oxygen inside. There's very low pH conditions in the bird's digestive system. Of course, you need to, depending on the animal, but in case of birds, they have a small stomach with all kinds of stones and grit in there, and then you'll be grinded, so there are all kinds of mechanical forces. And then, of course, digestive enzymes and potentially some bacterial fermentation going on. So looking at all these things that species need to resist. A few years ago, I wrote this review looking at how many species you can actually find if you collect droppings in the field. And basically, overall, for all those studies that collected droppings and found seeds or invertebrates, we found that almost half of all the droppings you would collect in the fields, imagining all these dots being droppings, and all the dark blue on the right would be droppings in the field that would actually contain an intact plant or invertebrate. And of those, about 1/3 would actually be viable as well. So when you're just looking at many droppings, for instance in the rice fields in Doñana, you can think that many of them will contain something at least. Then I also reviewed the number of, so the experiments that people had been doing until then, taking a bird, feeding it seeds or invertebrates, and watching the defecation and how many invertebrates or plants would be excreted. And they found that about 20% of all the seeds that you would feed to a bird, you would also retrieve in the droppings. And 20% of those would also be still viable. And that's made me think a little bit, because in the field observations, in the field collections of droppings, you find about 5% of the viability of propagules. So propagules is any dispersal unit of a plant or an invertebrate, I should mention. So these propagules. And then in experiments, you find about the same percentage. So this suggests some kind of underlying mechanism. And if you think about digestion of a waterbird or an animal in general, suppose an animal eats something and it wants to digest 100% of what it has ingested. It takes a very long time for the digestive system to process this until the last fiber they ingested is also taken up. So what animals do is generally lower this efficiency of the digestive system to about 3/4 of what they ingest, 80% of what you ingest, because then you can process much more food over the same time. Instead of working 2 days to process a certain amount, you can just, in a few hours, you can process 80% of that amount, and over time, your energy uptake over time is actually maximized if you don't go to this 100% assimilation efficiency. And this explains, in my view, I think this explains a large part why all these aquatic invertebrates and plants can still survive, because it's also not in the interest of a bird to digest all these things they eat. So that made me ask, what is the limit of what can be dispersed? Because if a bird doesn't want to digest everything it ingests, there might be tons of animals and plants that can be dispersed. So then we went, for my PhD, actually, when we went to this kind of extreme situation, we thought maybe aquatic snails can even survive this. So we fed these 4 different snail species. We fed them to mallards that we kept in cages. We force-fed the mallards with these snails. And indeed, we found that even these aquatic snails, one of these species, the Hydrobia Peringia ulfa, that can actually survive the digestion of a bird. So after 5 hours, we still found these living snails, suggesting that even something like a whole organism, a snail, can survive the digestion of birds. Of course, the percentage was very low, but then imagining many birds eating many snails, you could sort of calculate that it might have some impact. But the basic thing that it illustrates is that you don't maybe need these extreme adaptations to be able to be moved by by birds. If you just have adaptations that make you suitable for living in harsh conditions, such as a wetland, then that might already be sufficient for you to be transported by birds. And that explains why, a long time ago I reviewed this, but the many invertebrates and plants can actually be dispersed. Both aquatic and terrestrial plant species is becoming realized lately. And then for external transport, I think virtually all animals that have some kind of desiccation tolerance and don't die during the transport will be able to be transported. But then quantitatively, we don't really have a good overview of which species are able to disperse. Of course, there's lots of literature, but there's not a real quantification yet. And illustrating this point of that if you can just resist desiccation, then you will survive, is that we did an experiment where we deliberately attached some of these aquatic snails to the bills of birds, of mallards in captivity, with mud. Hypothesizing that if you are, as a bird, you're playing around in the mud, and then there will be lots of mud on you, and you fly somewhere else. Maybe some snails may be externally attached via mud. And what we've found is that if you look at the log of the snails attached over time, in the first half hour, you see that many of the snails fell off, which we also expected. But then, at some point, the mud was drying. And after this mud dried, the rate of detachments was much lower. And at some point, we went home, but the snails were still attached to the birds because of this drying mud. And that made us imagine that if a bird would be in the mud and it would not clean itself deliberately, which the mallards didn't do in the experiment, then it'd fly to another pond, and then when it resubmerges in the water in another pond, the mud will wash off, and whatever is attached, whether it's a snail or a resting egg of an invertebrate or whatever, then with this mud, many species can be dispersed. And then we did a nasty experiment where we tried to basically kill the snails by desiccation. That didn't really work because they could just survive, because they have adaptations for living in ponds that dry annually anyway, for instance. So desiccation is really not a big problem. So what I can conclude, or what has been concluded, is that quantitatively most like duck guts are more important than their feet, which Andy nicely showed a few years ago. So in terms of quantities, the internal transport pathway is much more important. And also for passerines has been reviewed. Passerine birds, it's also more important to what goes internally than externally. However, I think we still need to work a little bit on the qualitative aspect, because if you imagine the snails being attached to the outside, I don't think you need that many adaptations to be able to be transported on the outside. Like this picture illustrates, some species may be transported on the outside, which cannot survive on the inside. So there's something to still work on. And that includes, the dispersed species includes also alien species, which made me think quite recently. I synthesized like the aquatic studies that reported both the dispersal on the outside or inside of native and alien aquatic species. And of those published studies, we found that about 38% of the seeds that were dispersed were of alien origin in Europe. And more than half of the seeds transported on the outside were alien. So there's a strong bias in these publications, because they reported the alien and native status of the species that were dispersed. But it does illustrate that there may be some effect of alien species on dispersal by waterbirds. Because if you imagine that a bird encounters a native plant, but they're surrounded by alien plants, it may be eating seeds of alien plants instead of seeds of a native plant. So in that case, the dispersal potential of the native species by zoochory will be lower due to the arrival of the alien species. But this was basically a paper I wrote to make sure people realize this and to do more work on. So the next question was, which bird species are then important factors? What do we know about that? And then if you look at the which bird species we already know that may be important, it's like mostly dabbling ducks, diving ducks, and some of the coots. And there's quite some overlap in the number of species that they disperse, the kind of species that they disperse. So any invertebrate that might be dispersed by mallards might also be dispersed by teals, or the other way around. But in generally, the number of invertebrate species dispersed by each bird is quite, well, I think by now we know more, but it's quite high. And the number of plant species even much higher. But this also made us think what kind of birds would otherwise be dispersing all kinds of plant and invertebrate propagules. Well, so all these ducks and coots, but then maybe also some piscivorous waterbirds. So in a project with Andy, we tested a very old hypothesis that there may be some secondary dispersal by birds as well, so that piscivorous birds, fish-eating birds, would also be involved in transport, with the idea that seeds or invertebrates would be ingested by fish, which we know occurs, they just forage on this, but then in turn the fish may be ingested by a piscivorous bird. So we sampled cormorants. Cormorants eat a lot of fish, and then during the day, and then they fly to roosting sites on dead trees on the sides of ponds. And then in the morning, they produce this pellet. So all the fish they've been eating, they digest the fish parts, but then the fish bones and everything will be regurgitated. So we collected these pellets, a couple of locations in Europe, over 100 pellets, and actually in 1/3 of those there were seeds of aquatic and terrestrial plants. And in 1 out of 5 of these pellets, there were actually invertebrates. So imagine one of those pellets being produced while the cormorant is on the tree, and then it regurgitates the pellet into the water or on the land, and then the seeds may also be dispersed in a secondary way. The plant diversity that was found, we found about 20 plant species, of which most were terrestrial plants, actually, that the fish that maybe dropped into the water, and then the fish would forage in them, and then the cormorant would forage on the fish and then drop the pellet hopefully back onto the land. But also some riparian species and 5 fully aquatic species, including 6 species of plants that were not found before to be transported by dabbling ducks or diving ducks. So suggesting that dispersal by piscivorous birds may be an additional way for some plants to be transported that are not transported by other waterbirds. In terms of invertebrates, we found about 7 different taxa. So this suggests that, and then we tried to get an overview of how the relationships were. This is a sort of a network analysis between the plants in green, the plant species, and then the invertebrates in blue, and the fish in orange, to see if there were particular relationships between fish always eating particular plants and then the cormorants eating those fish. As a sort of a start of getting a better grip on how these secondary interactions may work. But this is still like much more work in progress. But it illustrates that there are many kinds of waterbirds, not only ducks, that are transporting seeds and invertebrates across these systems. So we're still sort of at the start of getting more grip on which waterbirds may be important seed dispersal vectors. So now I've shown that many invertebrates and seeds can be transported by many types and kinds of waterbirds. We can ask how far would they actually be transported? Well, that depends on how long they remain attached, but also how long it takes for them to pass through the digestive system. So if you remember this graph, the snails attached by mud, they just stand on endlessly until we stop the experiment. So we should redo the experiment and keep it going longer. But I think external transport can be quite extensive on migratory flights. But internal transport, it's a bit more. Well, we should study it a bit more. So this is a graph that is a combination of the decline in viability of seeds or invertebrates that have been eaten, and the time it stays in the digestive system. Because what generally happens is that seeds that are defecated later will have lower viability than seeds defecated after only 1 or 2 hours in the digestive system. But then there's a peak at about 4 to 6 hours after ingestion when most of the food will be defecated. So this graph sort of combines this. So then based on the hours after the ingestion of a propagule, how many of the viable propagules are released per hour. So then you see that after 1 hour, the average waterbird would shed about, in its droppings would shed about 3 viable plants or invertebrate propagules. But this is a model that should be still worked on and improved. But at least it illustrates how there's a relationship between the decline of viability and the number of propagules being defecated by birds. However, this retention time, how long a seed would stay inside of a bird, is hard to estimate from experiments with birds just sitting still in a cage. So if you imagine you eating a bag of candy in front of the television versus eating a bag of candy and running a marathon, the digestive system may be quite differently. And in order to disperse this candy or this seed, you need to move. So that made us think, what happens to the digestive system if the animal starts moving? Because so far all the experiments have been done with animals that are just sitting still in a cage, which is not really representative for movement of the animals. So we had a lot of fun. We built this flume tank, which basically is water circulated by outboard engines, silent outboard engines. Built some cages in there with sieves, so all the droppings that were produced, they would be sieved in water. And then we replaced mallards, ducks, we placed them in the water, and they had to swim, and they actually did so. So it was good. So they were swimming, so we had a swimming treatment here and a swimming treatment there, and then as controls, we had the mallards that were just sitting in the water. So they were still in the flume tank, but they were sitting on a mesh. So they didn't have to swim, but they were still in exact same conditions. And we used those as control mallards to see how the physical activity would influence the digestive system. And then we added a treatment which was resting in isolation, and it's basically the treatment that people are always using in the experiments like this. So we had a resting, and then wading, as we call it, like sitting in the water, just doing nothing, and then actively swimming. And what we found is that the retention time, so the time that markers, we fed them plastic markers, the plastic markers will be retained, and then the cumulative retrieval of those markers over time, we found that in the birds that were actively swimming, after about 4 hours, they're already defecating most of the markers that we fed them. But then those sitting still in the cage, most of the markers were retrieved after 6 hours. So that means that what we later interpreted due to the peristaltic movements, so the movement of the birds, would stimulate the processing of the food, the markers, through their system. Which means that if you're estimating that a seed would be transported a certain number by a bird over a certain number of hours, retained in this digestive system, this may actually be shortened if the bird is actually actively flying. So we did this experiment with plastic markers, and then we redid this with seeds to see if we found the same results. So, and then we again looked at this graph as a retention time and the cumulative retrieval of the open symbols are the markers being retrieved, and then the closed signals are seeds, and we found a smaller effect, but still the exact same effect. And then I put a little movie in for the because if you have a flume tank anyway, you might as well do some more experiments with that. And then you can find a fish in here. Because fish can also, in temperate regions and mostly in tropical regions, transport seeds because they eat seeds, and then the seed stays inside, and then they swim upstream in rivers. That is otherwise hard for plants to colonize. So then we had these fish, and then the water circulating, and then the fish were actively swimming. And then we had control fish in the aquarium. And the hypothesis was that maybe the system would be different for ectothermic animals than for endothermic animals. And this is exactly what we found. That is again the same graph, the retention time and the cumulative retrieval. The fish that were in low activity, so just in an aquarium, were shedding the seeds earlier than the ones that were actively swimming. So in the ectothermic fish, the common carp species, we actually found that due to the physical, the movement that they were actually swimming, the most of the blood flow was going to the muscles and not to the digestive system. So in this case, for the fish, they retained the seeds longer, and they may actually, by swimming, they might disperse the seeds further than previously estimated in the aquaria experiments with just resting fish. So the funny thing is that in the birds, the seeds are defecated earlier, but in fish, they were retained longer due to this physical activity. So that illustrates that we need to do more of these kinds of experiments, more species-specific effects of activity on how long they can be retained. And this is a dispersal kernel, so you can see the distance that seeds may be transported and the probability of dispersal, which we generated from this data in fish. You see that the peak is much higher for inactive fish than we estimate, and the tail is called the fatter tail of the dispersal kernel, so the potential to be dispersed in this active fish. So there's a kind of a change in what you would estimate fish to contribute to plant seed dispersal in particular river systems. So this said something about how far species can be dispersed and what we should still do with that. And then I didn't want to leave this study out on genetic diversity, which is quite old, but still it's fun because it has been done in Doñana. So it's the reason I was here in 2009. We looked at whether or not this dispersal by birds may actually have also consequences. Because of course there can be many seeds dispersed, but does it also affect the populations, or are seeds also dispersed by all kinds of other factors and ways? So then when I met Andy and Jordi in 2008 and 2009, I came to Doñana, and we were interested in this Visa acuta, this invasive snail from North America. And we sampled all these different ponds and the rice fields and some ponds there in Doñana, and looked at the genetic diversity. So these snails are very good at colonizing these zacaiones, if I pronounce it well. These little ponds that are dug for the cattle in Doñana, for the cattle to be able to drink during the dry season. So some of the ponds dry out really a lot, but still you can find these aquatic snails surviving in these ponds, which is pretty amazing. So we sampled in these ponds, we sampled the snails. And then also in the rice fields, which you're probably all familiar with, they're quite an abundant invasive species. And then comparing for about 200 something snails, different places, and then we had these fancy microsatellites, these that are now not so fancy anymore, but still they worked very well. And we compared these different populations, and we found that within the rice fields, these snails are all just basically the same genetically. Because the water is rising now and then, and there's lots of exchange of water flow, so the snails can just move by water. So by hydrochory, it's called like water movement. So water was an important factor inferred from this study. But then also we found that the ponds, the zacaiones in the north part of the Doñana National Park, they were quite genetically similar to the rice fields. Not so much as within the rice field, but still they were really similar. So made us think that potentially the seeds, the snails, will be transported to these fields and to these zacaiones here. But what we didn't, these populations here didn't really match the genetics here. So the birds would probably go more often to this side and to the river than to the far point. In the south. But then looking only within the park, we found that all the areas that we're able to access by cattle, we had some droppings of horses and dropping counts of activity, representing the activity of horses and cattle and deer and boar. We found that there's actually quite a lot of exchange between these zacaiones, but not when you look at the southern populations. So then we started thinking that maybe the large mammals that are like enjoying themselves in the mud of the Taxonomy that were running around and then running to another pond a kilometer away. Then the snails may be attached on the outside, and this could be another means of transport. So basically this study made us think about what potential factors these snails may be using. And of course we need like bigger sample sizes and do more of this work, but it still informs about what we think that these snails are using as terms of vectors. So to bring you to some conclusions of my past work before I outline some of the things I'm now doing. I think I've shown that waterbirds can disperse a large variety of aquatic organisms. Many waterbird species up to piscivorous birds may be involved in this. And this includes plants and invertebrates that are being dispersed, including also alien species. And then the reasons why this is happening is because many of those species have adaptations in wetlands to overcome desiccation, which they need anyway, and these harsh environments that you would generally find in temporary ponds, for instance. And then the passive attachment, you don't need much more than these adaptations that they already have. And because of this digestive trade-off in waterbirds, the waterbird is not interested in destroying everything it eats. So this way, also there's another reason for why so many species can be dispersed. And then internally and externally, the potential for many hours of transport is possible, which you could combine with the average flight speed of waterbirds of maybe 60 kilometers an hour, and then already you're quite easy to get a few hundred kilometers on a bird. And then we've showed some preliminary results on genetic diversity, where I do need to mention that this is all my own work and not so much the work of many other people that have been working on this. So I could make this talk like 3 times as long by including all the papers of other people on this, but I wanted to tell you today what I've been doing. So, and then a little bit about my own current work. So what I'm now trying to do in my Marie Curie project is getting a better mechanistic understanding of why particular invertebrates or seeds are surviving and why others are not, to see where the limits are of what species can be dispersed, and to see if we can estimate for which plants and invertebrates this is an important process and for which others it's not. So I'm trying to understand the mechanisms of this seed digestion of internal transport, which I'll say a little bit more about. And another aim of the Marie Curie is to merge the knowledge that we have on frugivory, so all the work that basically you guys have been doing on the fleshy fruits being dispersed by birds. There's a strong link to aquatic birds transporting all these seeds. So with the idea to see if there are any similarities and compare the evolutionary pathways of this. So this is also part of the reason I'm here. So if anyone is interested in discussing, because I don't know too much about this, so it'll be interesting to combine knowledge. And then in the end, once I have sort of been able to understand for which plant species it's an important process, combine this with biogeographical modeling to see if we can also see this a signal of zoochory in the plant distributions. So a little bit about the experiment I've been doing this summer, of which I had hoped to present some more results, but time is short. So what I found during all these experiments is that if you feed a bird with a seed and you put it in a cage, then one bird may be defecating a lot of seeds, but the other bird may not, because the digestive systems are quite different. So there's lots of individual variability among the different birds. Then you have these ethical considerations with these experiments where you don't actually want to force feed a bird and put it in a cage. It's not very nice to the bird. But also if you want to compare a seed species and to see if this seed or this plant or that plant can be dispersed, if you use 2 different ducks at the same time, then you have the differences between ducks. And if you use the same duck week 1 and week 2, you have also differences because the duck is also changed during this week. So that's why I came up with artificial digestion, to see if we can sort of change this, understand more of the physiological process of digestion, and to mimic the digestion of birds in the lab. So instead of using live animals, you just mimic the processes that they go through, and and no longer look at the bird as sort of a black box in which you put something in and then you get something out and you see what happens. Which I think can give me, but I'm looking at the data now, so I hope it will give me some grip on which plants traits, which seed traits are important for dispersal, and also calculate the maximum potential retention times of seeds and see how long they can survive, because you can just leave the artificial digestion process on as long as you want. So that's what we did. We built a setup where we basically simulated the process. This is you can imagine a duck around this. There's a esophagus and a crop in which the seeds the seeds or the invertebrates are retained. And then it goes into the propagules where there's digestive enzymes being added. Then it goes into the gizzard, and it's an important organ full of stones, basically where the seeds are being grinded and mechanically destroyed. Also under low pH and conditions with pepsine and other digestive enzymes. And then it goes into the intestines where more enzymes and things are, bacterial fermentation is added. So we simulated all this, and it all happens in a bird with 42 degrees, and then regulate the different phases of the process. And then we already see that these seeds are quite heavily damaged by the artificial digestion that we do. So if you look at some cypress sedge, for instance, you see that the seed goes in like this with a nice seed coat, and then it comes out like that. So you can also see exactly what's happening, which also may be helpful for identifying seed species that you find in droppings of birds, because then you know what they might look like after digestion, which is often quite different from before digestion. And then this milk partially also is destroyed, but some of these can still actually germinate because the internal structure of the seed is still there. And these iris seeds, they also lost their seed coat completely. And what I can already tell you now is that we already see some effects on germination, where these iris seeds have been like 3 weeks in the lab trying to germinate them, and they don't. But then you digest them for like 4 hours in this digester, and then they start germinating once you already basically in the digester. So it's really an interesting way to see the effects of gut passage or transport in the birds on things as time to first germination, but also we found susceptibility to fungal infections, which is also important after the seed has been found in a dropping. So we see some differences in control seeds and seeds that have been digested in fungal infections, where surprisingly, some of the infections were starting in the seed coats, while we actually thought that seed coat would be protecting the seed against fungal infection. So, well, I hope to be able to show more soon, because the germination experiments will be finished, I think, next week or 2 weeks, which will allow me to say more about which traits plants need for their survival, because we now have data for 48 different plant species so far. So I hope to work on that soon, and then also integrate that with the knowledge on Herbivorous and the traits that we know are important for dispersal of fleshy fruits. That brings me to the end of my talk. Thanks a lot for your attention, and I also want to mention that with Andy and Papatea, we set up this research topics, this special issue of in Frontiers in Ecology and Evolution, basically to stimulate people to do more work on Zoochory, which we are obviously interested in. And the papers will be soon coming out of that, so please keep track of that. And then thanks a lot for your attention. If you have any questions and would like the discussions afterwards, that is the whole purpose of this talk. So, thanks.